Unfortunately, my PowerPoint is not cooperating, so I have to use an old slide to talk about the differences between DNA versus RNA. Depending upon how you learn best, you can just write down these differences, or you can create a table that has DNA and RNA listed at the top and the differences along the side. The main differences category-wise are going to be the sugar used, the bases used, the shape, and the purpose. So let's start with the sugar used. So if you look at the name DNA and RNA, the first letter in each of those tells you what sugar is used in that particular nucleic acid. So DNA uses the sugar deoxyribose, whereas RNA uses the sugar ribose. And if you compare those two words, you'll see that deoxy means lack of an oxygen. So the big difference between deoxyribose and ribose is that one of them is missing an oxygen. But they're pretty similar in structure. When it comes to the bases present in each of these nucleic acids, DNA uses adenine, thymine, guanine, and cytosine. RNA uses similar bases, but it does not include thymine. Rather, it uses a base called uracil. So if you look at the pictures on the slide, for DNA on the left, you will see that adenine base pairs with thymine. Again, the more sciencey way of saying that is adenine is complementary to thymine. For RNA, if you look at the picture on the right, you'll see that where there's an adenine, it is base paired with uracil. So if I were to ever give you a random sequence of nucleotides and I said, hey, is this a strand of DNA or RNA? You would know the answer just by looking at the bases. If there's a thymine, you know it's a strand of DNA. And if there's a uracil, you know you're looking at RNA. Another big difference is the shape of these two nucleic acids. DNA is a double-stranded helix, whereas RNA has multiple shapes. There are 3 different types of RNA that we're going to talk about in this class, and each of them has a unique shape because it has a unique function. Remember, structure and function go hand in hand. That brings me to another category of differences between these two that I forgot to mention earlier, and that is the number of strands. DNA is a double-stranded helix, so it is composed of 2 strands of nucleotides. RNA is only a single strand of nucleotides. Now, sometimes this single strand of nucleotides will fold and the bases will interact with each other, like you see in this picture on the right. So that is a single strand of nucleotides that's kind of folded on itself to form its final 3D shape. So the function of each of these nucleic acids are related. So DNA is our genetic material. And that's really important because it tells us, well, it tells the cell which proteins to make and how to make them. We'll learn about that process when we talk about translation. RNA, again, there are 3 different types. So depending on which one we're talking about, there's a different function. We are going to talk a lot about messenger RNA, which is the directions that the ribosome and the cell needs to make a specific protein. And then we're also going to talk about transfer RNAs, which transfer amino acids from the cell to the growing polypeptide chain. So in other words, transfer RNA is going to be what actually helps build the proteins that are needed. So the messenger RNA will be the directions for the protein, and the transfer RNA has more of a role in actually building that protein. Okay, so those are the main differences between DNA and RNA. Let me know if you have any questions.